Okay, is it okay? Did you see my slide?
Yes.
Okay, thank you very much. My name is Idrissa, and I'm originally from Senegal in West Africa. I hold a diploma in as senior technician in biological analysis from Senegal, and I then moved to France where I obtained my bachelor degree in biology and also a master degree in microbiology. So after that, I pursue my PhD in molecular biology in Quebec City at Université Laval. And for this talk, I will present our study on the role and the importance of small non-coding RNA in the host pathogen interaction. So uh, just I will first talk about the class of small RNA in general with a specific focus on microRNA and tRNA fragment to define the framework of this presentation. Then I will present the synthesis of my main results. So throughout the presentation, I will evoke the context of the study and also all the rational foundation that led to our working hypothesis, and I will finish with the significance of the study in the small RNA research. So, to begin with this study, I first want to highlight that the concept of regulatory RNA were introduced in the 1980s with the progressive discovery of RNA that do not code for protein but play critical role in numerous biological process. So typically, small RNA or small regulatory RNA are non-coding RNA, less than 200 nucleotide, and most often they are involved in a post-transcriptional regulation. So generally, they are expressed under specific condition, and a major class of the small RNA act by base pairing with the target. It's the case for eukaryotic microRNA, most of the tRNA fragment, and also many bacterial RNA. So this can lead to the activation, but also the stabilization or sometimes the repression scenario. So in the context of this talk, I will briefly describe the biogenesis and the function of microRNA and also the tRNA fragment. So let's go with microRNA biosynthesis. It starts in the nucleus with the transcription of gene, which may be located in the exon or intron, and there's a primary transcript are cleaved by Drosha and its partners to give the precursor microRNA. And then the precursor microRNA are exported in the cytoplasm, in the cytosol, where it is they are taken up by another ribonuclease called dicer. Dicer is a key player in the generation of microRNA duplex. So the guide strand uh, of the duplex is assembled on the risk complex, and depending on the complementarity with uh, the targets mRNA, they can inhibit translation and or degrade mRNA, and also it can be a sequestration in a P body. So there are also some non-canonical pathway that can lead to the same goal. So actually, little is known about bacterial microRNA. The term is also controversial to talk about bacterial microRNA. So regarding tRNA fragment, they are fragments, so derived from the transfer RNA, and there's the biosynthesis of this transfer RNA is relatively similar in both prokaryotic and eukaryotic models. So they are transcribed into precursor tRNA, and with containing here in the 5' site leader sequence, and in the 3' site a trailer sequence. So the precursor transfer RNA are then matured in the 5' side, generally in eukaryotic model by RNAsP. And also it's the case, this is the case for bacterial model. So, and in the 3' side, the maturation is performed by endo or exoribonuclease, depending on the organism. So depending on the origin and also the size on mature or premature tRNA, the tRNA fragment are classified into several families. So we have TRF5, TRF3, we have also type 3 or type 5. So they are involved in regulating gene expression at transcriptional and post-transcriptional or also translational level, like microRNA. Um, However, here, what I wanted to highlight here, most of our knowledge on tRNA fragment over the last few decades were acquired through eukaryotic model. Little is known about the characteristic, the biogenesis, the distribution of tRNA fragment from bacterial model organism. So, how to identify this very small RNA? So, there are several techniques to identify there's non-coding RNA. Until the post-genomic era, the approach based on microarray screening and labeling were the main one used, but more recently, the advent of high throughput sequencing has changed the picture, and with RNA-seq in particular, because it was uh, a game changer. And also, there has been an explosion of knowledge of about small RNA, and RNA-seq become like a powerful tool for the discovery and the quantification of non-coding RNA. So uh, briefly, the RNA-seq experience is based on. Sorry, I forgot to translate my slide there in French. um, Briefly, the RNA-seq experience is based on three main stage. There are RNA isolation, library preparation, and sequencing. So in the RNA isolation step, there is one thing also that is important to highlight here. The experimenter decides on the size of fractionation. He can also decide of the enrichment of specific biotype. He can, for example, decide to do ribodepletion to remove all the ribosomal RNA. So however, what we see or what we observe is when building a library, it is not uncommon, or it's always the case to find that the lower limit of sequencing windows is set at 15 or 16 nucleotide. De facto, anything below 15 is considered as a random degradation product that cannot be explored. So all the bioinformatic platform also don't consider the RNA below 15 nucleotide. So our question is this a dogma or a well-founded scientific concept? That's what we are going to explore in this project. So to go back in the context, my arrival in the laboratory coincided with recent development of new project focusing on identifying or the characterization of exceptionally short, very small RNA, which we call it VsRNA for very small RNA. So, and this project was prompted by the discovery in our lab of a microRNA fragment, which we call it semi-microRNA, which were capable of modulating the microRNA from which it derived. So to find out more about the distribution, the conservation, and the role of the semi-microRNA, we extended the study to 5 different species and a lot of condition as you see here, and in the windows of 8 and between 8 and 13 nucleotides. So as you can see here, generally here are the windows of microRNA, and the common RNA size windows are here. And what you observe here is there is we confirm it here a relatively high abundance of 12 and 13 nucleotide RNA compared to the microRNA. So there's preliminary data are part of the thesis of one of my colleagues. So as I am my background are is in microbiology. When I arrived in the lab, I was interested to know if there are all the similar RNA, very small RNA in bacteria. So we therefore analyzed it over 6 bacterial strain in the 830 nucleotide windows by RNA-seq, and to characterize the profile of this very small RNA. So as can be seen here, the very small RNA was very abundant in bacteria and accounting for more than half of the read in several strains. So for this presentation, I will focus mainly on E. coli. So just to mention something, when I talk about very small RNA, I'm talking about RNA between 8 and 15 nucleotides. So by looking closer to the biotype of the sequence obtained by RNA-seq, we realized that they were mainly composed of tRNA fragment. And we then analyzed the secondary structure of this tRNA using RNA folds. And as you can see here, the tRNA fragment was thermodynamically stable with a stem loop structure which increase also the stability. And we have a reason to believe that they originate from specific processing rather than a random degradation product. And all the TRF that we identified in this biotype, they mainly aligned from the 5' side on the mature tRNA. So for the purpose of this presentation also, I'm going to focus on isoleucine TRF5X, which was one of the most abundant, the most redundant, and also the most stable TRF. For common sake, I make use 5X to refer to isoleucine TRF5X. So once we decide to focus primarily on E. coli, and we also isolated its vesicles called OMV for outer membrane vesicles. So for context, I landed in a lab also where the heart of the research has long focused on the study of microRNA transfer between cell and species using microvesicles. So as a result, I decided to include in order to benefit of all the tools that are available in the lab, I decided to include the OMV also in my analysis and experimental strategy. So what are OMV? So OMV are vesicles produced in all domain of life, and they are released in both gram-negative and positive bacteria. And why bacteria produce OMV? Mainly for three reasons. First, to increase the chance of survival. Two, second, to regulate interaction within the bacterial community, like biofilm, and also to promote pathogenesis by delivering some virulence factor. So um, in the laboratory, we have characterized the E. coli OMV by adapting isolation technique on a gel stained with Commercy blue, where OMV and also sample intermediate stage were migrated. The characteristic protein of OMV are clearly distinguishable here, the OMP-AC and the OMP-F. So under the electron microscope also, there is we can see clearly the OMV, which have a spherical shape and an average diameter of 180 nanometer. And we should point out here, we perform also a lot of other experiment to characterize the OMV, like dynamic like scattering, and then we isolated the OMV and extracted the RNA for the RNA-seq analysis. So here are the results. So by analyzing the distribution by sequence length and and by transcript, also what we observe here is just in E. coli and OMV, there are highly enriched in very small RNA, as you can see here. So we also note a certain selectivity in very small RNA loading within the OMV, as we can observe it here. So uh, by analyzing the proportion of the 5X in E. coli and OMV from the same culture, we observe there is a preferential packaging of 5X in OMV. So around 90% of the 5X are shorted, goes into the vesicles. So this should seem to indicate a possible role in intracellular communication, given the function of OMV that I presented earlier. So um, what are our hypotheses? First of all, I was interested in the dynamic of possible interaction between this very small RNA and the human cell on large exchange surfaces like colon. So we hypothesized that the community of bacteria found in the gut microbiota, they could interact with the colonic epithelial cell barrier by releasing OMV containing very small RNA capable of regulating the expression of recipient cell gene. So we use it HET116 cell line as a model. It is isolated from the colon of an adult human. So at the intracellular level, I mean, in the bacteria, the first question we addressed was whether the growth condition affect or modulate the level of 5x. So to this end, we first design and use a sensitive 5x specific RT-qPCR detection method because it's a very short RNA and it is hard, it is not possible to detect it with normal primers. And so in a rich and LB like LB medium, so um, what we see here, that the level of 5x did not seem to be affected by the growth condition. As you can see here, it was non-significant. But I wanted to, I don't show the data here, but we, at each time we measure the 5x level, we measure also the level of tRNA from which it derived to see the correlation between the two. So here, even the 5x level uh, show no change here. We observe that the mature isoleucine tRNA from which the 5x was derived or highly accumulated here in the stationary phases. So when we are in a low nutrient culture medium like M63, the level of 5x was significantly increased in above exponential and stationary growth phases. So here also, the isoleucine tRNA were reduced by 20% and here by 60%. So I'm talking about the mature tRNA from which the 5x was derived. We also studied the level of 5x depending on the temperature. So by lowering the temperature, there is no change in the level of 5x. But when we increase the temperature by adding 7 degree, 44 degree, we observe an increase in the level of 5x. Here also, the mature tRNA from which 5x was derived followed the same pattern as you can see here, the same pattern of 5x. So we then assessed whether the 5x level could be linked to transcription or translation. So inhibition of RNA synthesis by rifampicin and inhibition of protein synthesis by chloramphenicol did not affect at all the level of 5x. So therefore, the accumulation observed in the low nutrient medium does not result from a translation or transcription defect. And also complementary qPCR analysis, we perform it and we try to measure also the isoleucine tRNA. And here what we observe is there is a 20% decrease when you inhibit when you inhibit the transcription, and there is a 70% decrease when you inhibit the translation. But even if the tRNA, the isoleucine tRNA has shifted, there is no change in the level of 5x. That's why we said there is no correlation between tRNA level in the isoleucine tRNA level. So another question that arises or that emerged directly from this observation was whether the key tRNA maturation enzyme, such as the RNase E or the RNase P, are responsible for 5x generation, or if they in any way affected the level of these two enzymes, the level of of 5x. So we inhibited the RNase E and the RNase P, and we measured the level of 5x. Here, as you can see, the transgene inhibition of RNase E halved the level of 5x compared to the reference. And this result suggests that RNase E maybe play an active role in the biosynthesis and the turnover of 5X. The inhibition of RNase P, as you can see here, was have no effect on the level of 5X. So at the extracellular level, here in this section, we first studied the transfer of OMV into of OMVs into the HET116 cells. Here, the HET116 cells was uh, labeled with the red cell tracker, and the OMV with the green PKH67, and the nucleus with DAPI. So after 2 hours of incubation, as you can see here, a small proportion of bacterial OMV were already inside the cells, while the majority were still localized in the uh, cell surfaces. So after 18 hours of incubation, most of the bacterial OMV were internalized and dispersed in the cytoplasm. So between 2 and 18 hours, we observed a progressive and selective entry of OMV into the cells. So once we had demonstrated that bacterial OMV could be internalized by human host cell, we tested whether they contain could be transferred also. So in particular, the 5X, which is, as I shown it earlier, the most abundant very small RNA in bacteria and also in OMVs. So the cell exposed to OMV, so high level of 5X. As you can see here, this level decreased by third at 48 hours, but it remained very significant. So when we transfect directly the 5X into the human host cells, obviously we observe is here a marked decrease in the 5X at 24 hours, and at 48 hours, the level dropped drastically by 74%, and also it remained very significant. So once we know that 5X could be transferred to cells, the next step was to investigate the potential target. So for the purpose of this presentation, of course, we tested a lot of targets. I will limit myself to one target in particular, the MAP3K4, to illustrate the potential function of 5X. So the MAP3K4 mRNA display more than 10 potential binding sites for the bacterial 5X. Here in red, the potential binding site. So in the 3' UTR, we have binding site. In the open reading frame also, and also in the 5' UTR, we have targets. So I just want to highlight also the importance of the MAP3K4. It's an enzyme which is important transducer of multiple biochemical signal, and they are activated in response to how I can say, to inflammatory cytokine or some environmental stress, and also in throughout this interaction they may modulate the cell proliferation, the cytokine production, and also the apoptosis. So to test the hypothesis of a link between MAP3K4 and 5X, we first exposed cell to OMV, and then we measured the level of mRNA expression of MAP3K4. So we used LPS as a control, and as you can see here, the results showed that the presence of bacterial OMV um, increase the MAP3K4 expression at both messenger and protein level, as you can see here. It was significant when we incubate the cell with OMV. And here also at the messenger level, the MAP3K4 have a significant expression when incubated with OMV. So the exposure, the LPS did not show any results. So to ensure that there's observed increase of MAP3K4 can be attributed to imputable to the 5X. We transfected cell with either 5X alone, or a negative control of the same size, or also a cocktail of small RNA extracted from OMV, and 5X alone in the cocktail of small RNA were able to upregulate MA3K4 at messenger level. But as you can see here, there is no upregulation at protein level. So the bacterial 5X upregulate MAP3K4 expression in a second specific manner. Why we said that? Because we test also a lot of mutated 5X to see if the interaction is specific based on the sequence. So this increase of the MAP3K4 expression was um, surprising for us because we believe that a very small RNA would function like microRNA. So, and therefore it should repress the MA3K4 expression, but the opposite happened. So what we did next was to go back in and meticulously examine the many potential binding sites of 5X and MAP3K4. And interestingly, we found overlaps between microRNA and 5X. As you can see here, the microRNA binding site are highlighted in gray here. And the 5 most important 5X binding site are shown in red. So I said 5 because we have more than 20 potential binding site of the 5X in the MAP3K4 mRNA. So we cloned the entire 3' UTR of MAP3K4 into a reportogen system to perform luciferase EC. So here are the results. So in luciferase EC, we found that by introducing only the construct containing the MA3K4 3' UTR, we reduced the gene expression reporter the gene report expression by 60% in the wild type cell. So when we add the 5X, the signal is gradually restored. So 50 here and 100. But when we perform the same experiment in the mutated binding site of a 5X in the 3' UTR, so the restoration here is less evident, and the magnitude is less important here. So in the series of experiments that we perform in the lab, we also used HCT116 cell strain in which the dicer was deleted. So as I mentioned it in the introduction, the dicer play a very important role in the microRNA biogenesis. So as we saw in the introduction. So but when you remove dicer, um, more than 90%, 96% of the microRNA are depleted. So we use it as a control to see the potential interaction between, to confirm the interaction between microRNA and our 5X of interest. So in the HCT116, with no dicer. So the reported gene expression under the control of the MAP3K4 3' was reduced by only 10%. As you can see in it is totally different to what we observe here in the wild type cell. So by adding the 5X, the restoration is also very modest, about 15%. And here there is no significant results when we use it the version of plasmid where the 3' mutation have the 5X binding site mutated. So as the MAP kinase pathway are known to link the extracellular signal to fundamental intracellular process, we think that it was interesting to test to investigate whether the upregulation of MAP3K4 directly or indirectly trigger cell proliferation. So we used a colorimetric assay and to determine the effect of the bacterial OMV and the 5X on cell proliferation. So here, as you can see, the cell proliferation was moderately increased upon OMV exposure to bacterial OMV exposure, but here it was more significant with the 5X exposure. So finally, our results suggest that bacterial a very small RNA like 5X can be transferred to into human cells by OMV and constitute an important intracellular signal and in the bacterial host relationship. We have shown also that the transferred bacterial 5X upregulate the MAP3K4, a key player in the MAP kinase pathway, and the 5X activation of MAP3K4 promote cell proliferation. And also we tested, I did not present the data here, some inflammatory response and selectively trigger the P38 signaling pathway. So our study is the very first in E. coli to report the existence of abundant TRF smaller than 15 nucleotide found in OMV and assuming potential function in the host. So at the last part, I would say that the advent of high throughput sequencing technique like RNA-seq has greatly contributed to the to demystifying the non-coding part of RNA. And in our project, this technique has enabled to set light on the role of very small RNA in the host-pathogen relationship. And now it is clear that your we can say that the expression of the regulation of gene expression cannot take place without RNA as a functional entities. And consider that the protein play are very widely documented. By choosing to characterize the host bacteria relationship from the perspective of very small RNA, we aim to bring a new perspective to understand the pathogenesis of bacteria. And our project also remind the importance of revising some scientific concept of about the functional size of RNA, because this kind of dogma has delayed the discovery of a microRNA, the importance of the microRNA more than 50 years. So thank you very much. I would like to thank all my former colleagues in the Provo lab, and also thanks the partner who funded this project. So thank you very much, and I am available for your question.
Thank you, Idrissa. So, any questions from the panel for Idrissa?
Very interesting findings. I don't know a lot about OMVs or small RNAs, but I was interested in how did you come to the MAP3 kinase as being the sort of target of interest to, study further? I would guess small nucleotide sequences in a genome or may have a number of genes that to which they bind, and there seem to be a large number of binding sites.
Exactly.
MAP3 kinase gene. Are there other genes that have multiple binding sites that might also be affected and by this delivery of the 5X RNAs?
Yeah, exactly. So as I said in the presentation, we identified a lot of potential targets. So, but to filter this target and to choose the more important or the target that should be potentially be affected. So we use like three bioinformatic tool, RNA hybrid, some target scan, and a lot of other tools developed in the lab to be sure that there's potential target are the best. And with the RNA hybrid also we can understand or check the efficiency of the interaction between the 5X and the target RNA. But of course, we think that there are more than 100 targets that we identify that should be potentially studied, and maybe they should be affected also by the 5X. But we go just with our experience with microRNA to identify the tool developed for microRNA. We use this tool to identify the best potential targets, and MAP3K4 was one of the most important. That's why we focused on this one. But in the lab, we continue to study other potential target, and most of them were modulated by the 5X. Just to complete my answer here, the 5X is not it did not have an intrinsic gene regulatory properties, because when we use the dicer deleted cell, the 5X were able without competition to base pair with the triple UTR of the MAP3K4, but there is no change in the expression of the MAP3K4 because the 5X did not have intrinsic potential gene regulatory properties, but we think that it's a kind of modulator. So by coming and compete with microRNA, it may stabilize the expression of a lot of mRNA and maybe change so the profile of expression.
A complex realm to work in, for sure. Yeah.
Can I ask some questions?
Sure, sure.
Okay, so it's a very interesting talk, and thank you for presenting that to us. So I have just a few questions for the detail. First of all, you use E. coli, and did you get E. coli is a gram negative and just have the outer membrane vesicle. If you ever or anybody in your lab that tries Staph aureus, because that's a gram positive and they also have EV. And if you tried, what did you find?
Unfortunately, we didn't work with Staphylococcus, and we just focused on E. coli. Yeah, to represent, because in the hypothesis, my idea was just to, as E. coli is representative in the gut microbiota. So I was wondering maybe there is a potential interaction between this very small RNA and the colonic epithelial cell barrier. So I just focused on E. coli in the context of dynamic interaction between the two, but also we were not in a context of the Staphylococcus, as it is a gram positive, and also it need biofacility level 2. It was more hard to work with this bacteria, and the presence of Staphylococcus in the colon was not something normal. So that's why we focus just on E. coli to give the proof of concept. And then maybe the next step will be to focus on one of the gram positive. But we did not try anything about positive, gram positive, yeah.
And possibly in an epithelial cell, interaction study rather than colon for staph, of course, but yeah.
And so on that thought, if you happen to work with Staph aureus, would you be happy to pursue if you have a chance to work with Staph aureus?
For sure.
Yeah.
I'm curious to know what will happen with the Staph aureus, of course, yeah, because it's a different type of OMV. Maybe also the shorting, the selectivity, the selective loading of OMV will be different. So maybe we will not find the same pattern, and the most important very small RNA will be maybe different. I don't know, because yet we don't understand how the bacteria choose which of the RNA should be selected in the OMV and which one should stay in the bacteria, and why. We try to give an answer throughout this project, but it's not complete for sure.
Okay, that's very nice. And so yeah, that lead to my other question. Do you know how the RNA get loaded inside the OMV? Is that just a random event that when it's excised from exocytosis, it just grab a bunch of RNA, or did it have a certain mechanism that behind it?
Yeah, for sure, we believe that in our lab that this shorting or this selection in OMV is not something random. It is sequence dependent. There's it is not something that we study ourselves in the lab, but what I can find in the literature review is the sorting of RNA in the OMV is sequence dependent. So it's depend on the sequence of the RNA, and also in the context also of stimulation. Maybe if the bacteria are in a context of when we hit maybe the growth condition measure, we change the temperature. This stress can lead to a sorting of different type of RNA. So because we test a lot of condition in E. coli and OMV in a different condition, but we see that depending on the stress you apply on the E. coli, you don't get the same profile of very small RNA in the OMV. So it cannot be random. But in other case also, as the very small RNA are highly abundant, like the 5X is highly abundant in bacteria, so it's normal to find it also in the OMV, because the OMV production in bacteria did not depend because it is produced all the time. So it's normal just for, if I believe in probability, there is a high probability to have them in the OMV. But there is, I think that it's not random, but it's a mechanism behind it. Yes, as on the sequence. Yeah.
So on that thought, you're talking about environmental stresses, and you try the two major one, the growth condition, such as the nutrient and also the temperature. So what is the main difference between the M63 with the other minimum media like M9 or CDM? Do you have any idea? Is that the content of sugar, glucose, or amino acid, or anything like that?
Yeah, so good question. Here we use it because we have something in mind by using this measure. It's a low nutrient measure. It's about the access of carbon and also the glucose, the glucoses. In this project, when we perform the sequencing, the experiment of sequencing, we see that the RNase E, when it is inhibited, it can halve the level of 5X. And there is a link between the RNase E and the glycolysis pathway. So that's why we use the M63 measure to understand why RNase E, to have maybe a clue to better understand why RNase E are the one of the ribonucleases that are able to modulate the 5X. So our hypothesis was that as RNase E is implied in the glycolysis pathway, maybe it's if when we use this kind of low nutrient media, we will see the difference. But the answer was not clear because we have some result that is not going to support this hypothesis. But the reason that why we choose this measure is just to see in a low nutrient carbon measure what and glucose measure what is the behavior of the bacteria, and also how the 5X will be modulated. But we were able to choose another measure. There is no, the specificity, as I said, to choosing this measure is just related to what we know about RNase E and the glycolysis pathway.
Yeah, okay,
But
thank you.
It should be interesting to test also another measure for sure. Yeah.
Okay, so one last question. You got your master degree in France, right? In Paris?
Yeah.
What university?
So I get my bachelor degree at Université Orléans and my master degree at University of Toulouse, Paul Sabatier.
Oh, okay, because I come from University of Paris, Orsay Center.
Okay.
On peut parler
It was in
français. Okay, thank you
Avec
very
plaisir.
much.
So I need to improve my English. So I'm a French speaker, so that's why. So if something is not clear, don't hesitate to.
In French.
So Idrissa, so I appreciate your background in molecular biology and microbiology. So, and because the research here for sure is relevant to your background, but also quite different because we are very much translational. So if you come here, maybe you have to be involved in some some engineering work with engineers. So how comfortable are you going to be with that? So and what has motivated you to apply for this position? Yeah. Can you comment on this?
Yeah, I think as a aspiring researcher, I'm, very, I know, I mean, I'm very interested to work in a translational group because it's a very good occasion to learn from, people working in other fields, first of all. But the thing that uh, bring me to this project and apply, it's first of all, it's a challenge because I see that it's something new because I know that the antimicrobial resistance is the thing is a new subject that will be our, I mean, uh, how to say fléau in English. I mean, it's the challenge for humanity. I read somewhere by 2050 there will be more deaths related to this problem than cancer. So I think it's a big challenge for humanity, and by seeing people are working on this project by using different approach, I was just curious to understand how it works, like based therapy. So how it work, what is the mechanism behind this project? So is it a potential preventive effect? So when I see the job offer, I a lot of questions rise in my mind, and I was interested to learn more about this, and that's why I apply. And I think to see that there is possible of non-invasive method to use it to to treat antimicrobial resistance is something that is very interesting and new. And I know that also there is a controversial aspect of this approach because people don't really understand how the mechanism behind the light-based therapy. And I think that being part of this project and contributing to bring information and scientific concept clearly established, it's very interesting for me. So that's why I apply, and I will be very happy to work with engineer and a lot of people that work with in project that is nothing to do with what I know. So it's a good occasion to see the project in a different perspective and to learn also. But I'm I'm a student, so still a student, so I need to learn. So it's very interesting for me to be there in this kind of translational research team. So
Thank you for all the points. So in your application package, you mentioned you would like to live in Boston, right?
yeah,
You would like to move to Boston and live here, right? So another goal of you, right? To apply for this job.
exactly.
Okay, any other questions for Idrissa from Carol Dazio?
Hi, Idrissa. Nice to meet you. Very
I
nice presentation. I'm
Thank you.
excited to know more about you. What is your expectation? This is a completely different field for research from what you are doing now. And I want to know, how do you think the training that you are going to be here will help you in your future? What is your goal for the future? And because this is a completely different field, and you need to know if this will adapt it for your goals. And I am excited to know about you have the expertise about microbiology, and do you have some specific disease that you would like to treat with light? I also want to comment that we are working a lot of translational research, and I'm very excited to work with you in translational studies, doing a lot of things, learning things every day, and I want to listen about you and know more about you.
Okay, thank you for the question. So, I think that after my PhD, I was working on a very fundamental research project. So, uh, the thing that I present here is very fundamental. So, after this 6 year of PhD, I was interested to be in something that is more applied science. So that's why these kind of projects have a good interest for me, because it's a good opportunity to be directly involved in something that is really applied, directly applied, so that we can test it and we can understand, monitor, change the parameter and see if it work or not. So this is the one part of the interest of this project. And also I think that I don't know exactly what I will be in 5 or 10 years. The only thing I know and the only thing I want is to learn a lot of and enough to be ready for anything in the future. So being involved in this kind of project, so for sure, will increase my resume. Also, I will defend that I have uh, a large spectrum of knowledge related to different subjects. So I think for sure it will be a gain for me. It will be a plus for me. So uh, this is what I can say about it. And I will be very, as I mentioned it earlier, uh, I'm not afraid about being in a translational research or being in a very new project, because when I arrived in the lab here in Quebec City, they were they never use bacteria. The lab focused only on eukaryotic cell. They don't know. They don't have medium for bacteria. The only focus is eukaryotic models. So that's why, as you can see in the presentation, I don't focus more on the potential function of 5X in the bacteria. I mainly focus on the dynamic of interaction between the bacteria and the human host cell, because it was the only way for me to use what they have as a tool, because they don't have any tools for bacteria. I build I knock out the gene if I have to knock out it, and I do it by myself, and I try to modify or to adapt the project based on what I know in microbiology to the tool available in the lab. So it's something very exciting and challenging, and I think that I will have the same feeling by being involved in this kind of project. So something totally different. You come and just with the basic science, learn fast and try to be involved and do your job as much as possible. So yes.
Any more questions from the panel? Okay, if there's no more questions, so I would like to wrap up today's interview. Thank you again, Idrissa, for your presentation, also for your interest in this position. So we'll be in touch regarding the next step. Yeah. Okay.
Thank you very
Thank you,
much.
everybody. Yeah.
Thank you for giving me this opportunity to talk to you. Merci beaucoup.
Okay. Bye, everybody. Yeah.